I find a lot of the sort of wilder stuff that's going on really exciting, and I'm just really fascinated to see how it pans out. And I think the treatment I'd put most in this category is cellular reprogramming, because I often say it feels like it's fallen through a wormhole from the future.
So hello and welcome to the Cheeky Science Show, where in this video I had the great pleasure of speaking with Andrew Steele, author of the book Ageless, and full-time science writer and presenter based in London. Andrew previously obtained a PhD in physics from the University of Oxford before steering towards computational biology. Here we discuss some of the fascinating discoveries in his book Ageless and his thoughts on how computational biology can be applied to aging research. I hope you enjoy our conversation. So, hi Andrew, thank you for joining me today and welcome to the Cheeky Science Show. It's great to have you on today.
Thanks for having me. It's a pleasure. I'm looking forward to it.
Thanks. So my first question, before we go into the details of your book and your interest in the aging research, his first question is just to get a better understanding of your background, which I believe was a PhD in physics. So I was just wondering, what initially drew you to the area of physics, and then what kind of led you from physics into the more computational biology routes?
Yeah, it's been a bit of a journey, and I guess like an awful lot of scientists, actually, my sort of science origin story is back when I was, my mum says, about 7 years old. I'd look up at the sky, I'd look at all the constellations up there, all the stars, and you sort of ponder my place in the universe. And I think about that age, I decided I wanted to be a theoretical astrophysicist. And slowly but surely, my career has sort of diverged away from this sort of incredible, abstract, fascinating big question sort of end of science into the more practical, trying to help people end of things. So I ended up, as you say, I did a degree and a PhD in physics. I worked on magnetism and superconductivity during my PhD. But toward the end of my PhD, I was trying to look for a way that I could use my career to make a difference in the world. One of the things I considered, for example, was becoming a climate physicist, which would have been a bit more of a conventional transition, trying to use those skills, to understand and hopefully tackle the problem of climate change. But at that time, I started reading about aging biology. And actually, what I normally tell people, which is a bit of an oversimplification, but not much of one, is that I changed career because of a graph. And that graph is the graph of human mortality risk as associated with age. So obviously, I think everyone watching this channel will be familiar with the fact that humans' risk of death doubles every 8 years. And as a physicist, you look at that and you think, wow, this is just this staggering thing that applies across human populations. It applies across time. I can take basically any human from any place on the earth at any time in human history, and their risk of death will be different depending on when they lived, but it'll still have this same sort of ticking clock, this doubling every 8 years. And so you look at that as a physicist, you're trained to see patterns in incredibly complicated phenomena, and you think, wow, is there something going on here inside our biology that's causing this fundamental, incredibly constant ticking clock? And if we could go in there and intervene, could we do something about it? And so then I started reading about, things like calorie restriction and rapamycin experiments, just getting quite big around that time because that was when the interventions testing program results had just come out about that. There seemed to be loads of exciting stuff going on, but I did sort of have this niggling doubt at the back of my mind. I stopped studying biology formally at GCSE, so that's 16 years old for anyone who's not in the UK. And, obviously therefore I'm scratching my head, maybe I'm a physicist doing the classic physicist thing of looking at a field and thinking, we can just go in there and nail that. We can just, draw a graph and understand the whole thing. Actually, maybe it's just a bit more complicated than that. And that's why biologists aren't concentrating on it as much as I perhaps thought they should. So I decided I was going to try and become some kind of biologist. And there are various different ways as a physicist you can try and apply those skills to biology. A common one, which I've got a few friends who've actually done this route, you can take the very practical lab bench experimental skills and become something like a microscopist. So you can use all the optics that you learn as a physics undergrad and during your PhD and use it to develop new microscope techniques or something like that. And I sort of thought, really, what I want to understand are the big biology questions. And although obviously it's hugely, hugely important because these new microscopy techniques and so on allow us to do entirely new kinds of science, they allow us to literally see things we couldn't see before and discover new stuff as a result. I wanted to do something that gave me a bit more of an overview. And so that's why I thought that computational biology and genomics and that kind of thing was a bit, more than niche for me. You can still use the analytical skills. Obviously, I'd learned a bit of coding as part of my PhD as well, and take all of those skills and apply them to biological data instead of physics data. And then I just felt that would give me a bit more of a sort of bird's eye view of the field and hopefully, try and understand whether aging was indeed something we could do something about. And if so, how, and try and contribute to that.
Yeah, that's a really good answer. And I was actually going to say, I'm actually a bit jealous that you've done a PhD in undergrad in physics, because obviously I haven't done physics since A level. And obviously there's so much overlap between the different disciplines of science. Even in biology, there's now processes such as mechanotransduction and electrical signaling, and actually having that kind of physics understanding could be really useful to actually understand some of these more complex phenomenons in biology. So I was just wondering, is there any way you could elaborate on how you think your skill sets in physics help you to apply that to biological research?
I think at the moment we're at a point where actually a lot of the physics stuff is a bit bleeding edge in biology. I think quantum biology is a really good example. I think it's going to become abundantly clear over the next 5-10 years that quantum processes are going on in biology, because evolution has had billions of years to optimize our enzymes and all different stuff going on inside our cells at an incredibly detailed level. It'll be frankly astonishing if it wasn't using quantum mechanics on some level to optimize all those different processes. But the trouble is we are right at the bleeding edge of whether that's actually useful. I mean, are we going to find out that some quantum biological phenomenon is a 10th or an 11th or a 12th hallmark of aging? I guess we'll find out. But at the moment, I think that, like I say, a lot of the physics stuff is about technique development. And actually, I ended up basically not using my physics knowledge at all. I think it's quite cool just to have a really general grounding in science. And another thing I could have gone into because of my magnetism and superconductivity background is something like MRI imaging, which obviously depends on these enormous magnets to build detailed pictures of what's going on inside the human body. And I think it is really important just to have a bit more of a general scientific background to understand how different fields work, and perhaps just look at things with a different perspective. Because I think although I didn't have that biology training, perhaps in some ways I didn't have that baggage either, so it allows you to look at things in a different way. So I don't really think there's like a simple formula. I certainly wouldn't recommend, if if your passion is aging biology, I wouldn't recommend that everyone go away and do a physics PhD first. It's a bit of a circuitous route, but I think those things, and having a fresh perspective on stuff can definitely be an advantage as well.
Well said. And so to go to your book Ageless, I'm going to take one quote from it, which I think kind of hints at your more physics understanding, which is you describe life is under no thermodynamic obligation to age. So I was just wondering, could you break down that statement for us?
Yeah, and I think there are two ways of looking at that. So people, when they talk about aging, and I think even when you think about aging, we often think of it as just a natural process of falling apart. And you can really see why this would be. The science of thermodynamics was born in the sort of 19th century primarily. People were looking at steam engines. They were trying to understand the actual physical processes going on inside them. And, machines like steam engines, they rust, they decay. Machines in factories, they wear out as different components get, basically friction rubbed against each other if you don't lubricate them properly, etc. And it just really seems like, why would humans be any different from the machines that we can build? Well, the answer is, of course, they're biological, and so they've got these incredibly complicated self-repair mechanisms going on inside them. But it might be that perhaps there is this problem of thermodynamics. So the second law of thermodynamics is that entropy will tend to increase. And what's entropy? Well, it's that's a whole sort of video interview in itself. But entropy basically is a measure of disorder. It's a measure of things becoming more disordered, going from an ordered state into one where things are a bit of a mess. And so the second law states that entropy will always increase with time. And what that means, therefore, is that you expect an ordered physical system, a beautifully ordered human body, for example. Just holding myself up here is the most beautifully ordered example of humanity by doing that hand gesture. But my body should, under the second law of thermodynamics, you might expect it to gradually deteriorate, because at some point, those repair processes aren't going to be able to keep up with basically the endless march of thermodynamics. The problem is that people who say that neglect the second half of the second law, which is that entropy tends to increase in a closed system. And what does a physicist mean by a closed system? They mean a system that can't import energy and can't export any of that entropy or disorder into its environment. And clearly, living things aren't such a system. We can eat food, which is obviously the way that we get our energy, or if you're a plant, you can photosynthesize, you can use the light from the sun to generate energy yourself. And then we can export that entropy through a variety of different means that I won't go into too much graphic detail about into our environment. So we can get rid of that disorder, we can get rid of waste effectively. And so there's no sort of physics reason why it would be that we should age. And so that sort of demolishes that argument just by looking at it from a purely theoretical standpoint. I think the thing that's more compelling to me, though, actually, is then to go to the biology, because you do get to a point where you don't care about the physics if you've already got a biological example where this doesn't happen. I already mentioned that humans' risk of death doubles every 8 years. There are loads of creatures out there whose risk of death doesn't double at all. It stays flat when they become adults. And the reason there's a tortoise on the cover of my book is that giant tortoises are one such creature. They've got a risk of death that doesn't change with time. They don't get any more frail depending on how old they are. They even stay reproductively active until they're late in life. So in a very real sense, they don't age. So not only is not aging not against the laws of physics because it doesn't defy entropy, it's also not against the laws of biology, because we can point to tortoises and salamanders and fish and naked mole rats, all these animals out there in the world that don't seem to age either. And so I think there's this sort of, I can understand where this fatalism comes from, because it does seem that aging is inevitable. And we aren't really exposed to these creatures. There are obviously a handful of people who have a pet tortoise, but they probably haven't done a detailed demographic study of 1,000 pet tortoises in order to come to these kinds of conclusions. So we naturally assume that everything ages, but actually it's not that inevitable at all. And that's, I think, one of the most exciting things about this field.
Exactly, that's really well said. So, talking more generally about your book, what kind of motivated you to write the book in the first place?
I was inspired to write the book actually by my work as a biologist because I found that a lot of the biologists I interact with during my work, I'd often know more than they did about aging biology. And that isn't because I'm some kind of genius, again, not wanting to sound like a physicist who's barging into the field and feels like they suddenly know everything. The reason was that, I'd meet people with incredible degrees from incredible universities and they'd never have had a lecture on aging biology. There's not a single page in a lot of biology textbooks about aging. And that's just sort of staggering to me because it's one of the most universal processes. We've just heard it's not quite universal, but a huge number of species age, and it's evolution is perhaps the most universal phenomenon in biology, but aging is definitely up there as one of the others. And I think it's this sort of strange phenomenon that aging has been quite a small field historically, perhaps because of what we were just talking about, actually, because people have thought it's this inevitable process of falling apart that we can't really do anything about. Maybe it's just too complicated for biologists to take a serious interest in. And because the field was small historically, other fields have been larger, it sort of creates this vicious cycle. And you find that, if you're never lectured about aging biology, you're never going to get excited about it. So you're not going to apply for a PhD in it. And even if you want to apply for a PhD in it, there just aren't that many labs with PhDs to apply for. And that means that, when you get to the end of your actual PhD in cancer research or in virology or whatever it is, well, what are you going to do? You've already got a few papers, you've already got a bit of experience in that field. You're going to go on and become another cancer researcher or another virologist yourself. And that means that when you're lecturing your students and inspiring the next generation, you're not going to be telling them all about aging biology, are you? You're going to be telling them about your field of expertise, and so on and so on. This sort of perpetuates this cycle, which means that aging biology, and I assume therefore other neglected fields of science, remain neglected purely because they never quite gain this critical mass. And I really think I sort of diagnosed that pathology, and I thought that I want to write a book for a variety of reasons. Basically to convince lots and lots of people about how exciting this field is. And obviously, among those people, I count the general public, because I think that people just don't understand this science. They think it's some kind of sci-fi kooky weirdness when you talk to them about it. In that same sort of category, I'd put politicians and policymakers, the people who are funding the science. They don't realize, every politician knows a friend or a relative who's died of cancer. So cancer research gets comparatively within science, a larger slice of the pie, perhaps. Whereas people don't think you can die of aging. They don't see cancer as a disease caused by the aging process. So again, you've got to try and communicate to that group, what aging biology is all about. But then beyond that, I needed to communicate to the biologists as well. I need to show them that this is a legit field. This isn't some sort of cranky snake oil, people trying to live forever nonsense. It's actually genuinely hard science that, that works. We've got, dozens and dozens of labs around the world showing really promising results in this now. And actually, I then went on to marry a doctor. And I found that doctors also don't know much about aging biology. To the extent that they're taught about aging, when they have their lectures on aging, it's about, well, it's very complicated to care for these geriatric patients. They've often got multiple different diseases, so you have to think about that. There are social problems because, perhaps they're living alone at home, and so that complicates things. They're taking, 5 or 10 different drugs. So if you want to prescribe them a new drug, you've got to check how those drugs are going to interact. But nothing on the fact that, during my wife's career, she's quite likely to have to prescribe people these first generation of anti-aging drugs. It's not wildly outside the bounds of possibility. And yet when she first met me, I was crazy. She thought, what is this sort of mad, kooky sci-fi anti-aging stuff? And so in order to get the message out to that whole range of different audiences, I just thought, I've got to try and do something about this. And that's what made me decide to write a book.
Yeah, well said. And I have to say, having read your book, it is very well explained, and I think it's very accessible to people from any background knowledge. So yeah, congratulations to you for that.
Oh, thank you.
And so in your book, you discuss a lot of theories of aging, and to begin with, you speak a lot about the different evolutionary theories of why we might age. So I was just wondering, having done all this background reading to write your book, what theory do you think makes most sense to you at the moment to explain why that humans age compared to these other organisms you've discussed that seem to not show phenotypes of aging?
I think the most important thing to remember about the evolution of aging is this sort of strange paradox at its center, which is that the reason that animals die of aging is because ultimately they die of other things. So to sort of unpack what that means, if you find yourself in a very, very high-risk environment, let's imagine you're an animal like a mouse, and mice live, maybe 6 months or a year in the wild, so they've got very short lifespans compared to an animal like a human. And what that means, basically they're at high risk. They've got cats chasing after them all the time. They can get infectious diseases. They're tiny little animals, so they can literally just die of exposure, it's called. They can get so cold on a winter's night, they just freeze to death. And so there are loads of different ways that a mouse can meet a sticky end. And so if you're evolution, looking at this scenario and trying to design a mouse's sort of reproductive strategy, you're going to say, make a lot of babies and make them as fast as possible so that your genes have been passed on to the next generation. You have to make lots of babies because lots of them are going to die in horrible ways as well, unfortunately. And so ultimately, their reproductive strategy is sort of have a lot of kids and have them fast. And if that comes at the expense of lifespan, say that means that they put a bit more energy into reproducing or growing more quickly, and a bit less energy into creating incredibly robust anti-cancer defenses, for example, that are going to allow a mouse at the age of 2.5 to not get a particular tumor, well, they're just not going to bother with that, are they? Because they'd rather spend that energy on reproducing, because all the mice in the wild are already going to be dead by the time they get to the age when they're going to be experiencing age-related problems. So that's sort of one side of the coin. And that's why the evolution of aging, this sort of drive to not bother with any kind of long-term preservation strategy, evolves in animals that have a naturally short lifespan for other reasons, because they have external causes of mortality. If we then look at the other end of the scale, a nice example of this is bats. So bats, they're also, they're mammals, they're quite closely related to mice in the sort of evolutionary tree. But the key difference between a bat and a mouse, even though they're about the same size as well, a bat can obviously fly. And it's not just because flying is brilliant that these things live a lot longer. But if you look out in nature, then a bat can live to maybe 40 years, I think is the record. I think it might be 41 years old. So why is it that bats can live so much longer? It's because they're in a much safer environment, precisely because they can fly. They've got far fewer predators. And so, that means that they can afford to put more energy into getting rid of cancer, into, trying to keep their bodies running for a much, much longer period of time. And so that entirely changes their evolutionary strategy. And this manifests in a few different ways. It can manifest by something called mutation accumulation theory, which is something that happens over evolutionary time, where mutations that are really bad in late life can accumulate in an animal like a mouse. But a bat would deaccumulate those mutations. It would try and get rid of anything that might kill it. You can then have unfortunately, all the ideas in aging biology, aging evolution have really, really terrible names. Perhaps one of the most explanatory theories is this thing called antagonistic pleiotropy. So let's unpack that. Antagonistic obviously means that these things pull in different directions, and pleiotropy is the word that biologists give when a gene has more than one characteristic. So, so it can do different things at different times or in different situations. And so that then means a gene that might be useful for gearing up rapidly to reproduce in youth actually causes some kind of problem later in life. And that's, again, it really makes sense for the mice. They might have some gene that makes them grow more quickly, but makes cancer more likely as they get older. And so, again, you can see this antagonistic thing, the pleiotropy, the gene behaves differently depending on which part of the life the mouse is in. And then finally, the other theory that I discussed in the book is this thing called disposable soma, which is just the idea that, again, they've all got awful names. Disposable obviously means what it means. Soma means the sort of body. And that's just the sort of the biologist's way of saying evolution doesn't care about you. It cares about your genes being passed on to the next generation. And so, again, if you're apportioning energy between looking after your body and looking after the next generation, well, most of the time, particularly in a dangerous environment, you're going to put it towards the next generation rather than yourself, which sounds all very altruistic, but ultimately, of course, leads to us aging. So how is it, given all of these things, I've just described 3 different mechanisms by which animals can evolve in order to age. And it's not really on purpose, it's sort of an accidental byproduct of the fact they get killed by other things, but nonetheless, how do you get animals that don't? I think it really varies depending on their reproductive strategy. And I think the clearest example of this is in fish. So I just mentioned that, mice love to have loads and loads of kids when they're young, but fish have a very, very different life cycle. And that's to say that a huge amount of the reproduction by fish is done by massive old fish. And there's a great acronym for this. It's BOFs, which is big, old, fat, fertile female fish. Let me get that right. So BOFs. And these fish, they're massive underwater matriarchs, basically. They produce far, far more eggs than younger female fish. The eggs are of higher quality, so they're more likely to grow up into healthy fish themselves. And that means that a huge fraction of the population's reproduction is done by sort of this comparative handful of really old female fish. So evolution suddenly has a huge incentive to make sure that old female fish don't just die of cancer or die of something else. It wants to carry on, keeping them alive in order that they can carry on churning out babies. So again, it just completely changes the calculus. And it's obviously scientists are slightly squirming in their seat now because I'm giving evolution a sort of a motive. I'm saying evolution wants this and wants that. But you can see in that situation why evolution would want to keep that older fish alive. And depending on your life course, evolution doesn't care how long you live. Evolution will do whatever it can to maximize the propagation of your genetic material onto the next generation. And so if that, in the case of the fish, means that you might become negligibly senescent, you might even become negatively senescent, so you might have a reducing chance of death with time, if that's what evolution thinks is important. On the other side of the coin, if you're a mouse and you're in a really dangerous environment, you're going to crank out kids and you're going to age very rapidly. So I think basically evolution doesn't care how long you live. It will just try and maximize the number of kids you have. And unfortunately, that means that we are one of those animals that does age.
Yeah, well said. I was actually going to ask you about the buffs, because that was something I hadn't learned about before. And it's kind of fascinating to see examples where you seem to get promotion to just enable to increase fertility, but also to reduce signs of aging, so that these older fish can also remain reproductive as well. So that's obviously a really interesting example. And I think understanding different organisms in general is very useful because we can always use insights from different species to develop even human therapeutics. So kind of shifting gears more to the molecular underpinnings of why we age and how we can do something about it. You mentioned a nice example in your book about lipofuscin and how maybe it's not lipofuscin. You mentioned examples.
I say lipofuscin, but again, it's one of those words that you read it and you almost never actually say it.
It's true, yeah.
So we can go with one or the other. I'm happy.
But you mentioned how there's actually some enzymes. So this is something that accumulates in lysosomes, but it's something that our bodies can't naturally degrade. We have no enzymatic activity to do it. But there's been some recent research that you describe, and I'll let you explain it in more detail, but it's the idea that we can effectively use information and biology from different organisms to help treat aging in humans.
Yeah, and I think there are just some really fascinating examples of trying to draw in enzymes, because there obviously are creatures that can break down lipofuscin somewhere, because otherwise, the entire surface of the Earth would basically be just covered in huge piles of this stuff. But something must be degrading it in the environment. So we can look to things like soil-dwelling bacteria that might be able to degrade it. I think the one that I found most fascinating, and this, I'm not sure whether this is technically lipofuscin, but it's about oxidized cholesterol that builds up in the lysosomes of cells that are part of atherosclerosis. So when you're, basically the furring of your arteries as you get older, the cholesterol gets gobbled up by these immune cells, but unfortunately they can't really do anything with it. They just tuck it away in their lysosomes. It often gets oxidized. It often gets all kinds of other reactive stuff stuck all over it. And we were scratching our heads about how on earth we could possibly, get rid of this stuff. It's very, very hard to degrade, which is obviously why they can't do it. And what the researchers found was there's been this long-standing mystery about how tuberculosis bacteria survive hiding out inside immune cells, potentially for years or even decades, because tuberculosis is an infection that can go dormant and then come back out at times of stress. And what they found was that the tuberculosis bacteria were surviving by using some kind of cholesterol metabolism. So they were effectively digesting the cholesterol they found inside these immune cells. And so what the scientists did was they extracted obviously the enzyme that was responsible, inserted it into some cells in the lab, and they found that it did indeed successfully break down this oxidized cholesterol. The only problem was that the byproduct it created during that reaction was itself toxic to the cell, so it wasn't a sort of long-term solution. But you can see how with a bit of tweaking, that's something that we could use to attack the root cause of atherosclerosis, which is the leading cause of heart disease, basically is the cause of heart disease. And that's really exciting because the way that we currently deal with heart disease is just to try and reduce the level of cholesterol in the blood, which does slow down the accumulation of these plaques, but can never ultimately stop them. But if this can go in there and clear out the stuff that's causing all these immune cells to die and causing these plaques to form in the first place, then that's one of the dreams of aging biology, aging research, is to go in there and actually solve these problems before they accumulate to the point where they cause disease, rather than just slowing these things down. So I think this is one of the more sort of exciting but speculative areas of research that I talk about in the book, and I really hope we can sort of take some insights from other species and crack it.
Yeah, so you raised a really interesting point there, that there are kind of, I guess, two main strategies for trying to treat aging. One is trying to, the more like preventative strategy to target the root cause and prevent the bad things from happening. And then the alternative strategy is to kind of clear away the damage that's already accumulated. And so I was wondering if you could maybe talk more about the latter strategy. As do you think that that's probably the more likely therapeutic outcome we'll see first before we see these more preventative strategies?
I think it's an interesting question, and I think that often it's perhaps overplayed in the sense that there are maybe some things that fall really clearly into one category or another, but there are lots of things that seem to probably do both. Like if you think about rapamycin, this is something that might be characterized as something that just slows down the aging process. It's a drug that goes in, it clogs up an enzyme that effectively is part of the response to calorie restriction. And we know that if animals calorie restrict, if they eat a lot less, they can live a lot longer and they can do so in good health. So by jamming up this particular enzyme, it tricks your cells into thinking that you're eating far, far less than you are, but in doing so, therefore activates some of those longevity pathways that seem to be activated through calorie restriction. And you might think, therefore what you're doing, calorie restriction doesn't allow animals to live forever. It just slows down their aging. It doesn't make them negligibly senescent at all. So, maybe that is just a drug that slows down aging. But actually, rapamycin seems to have some effect on cellular senescence as well. It seems to get rid of senescent cells. And you might class getting rid of senescent cells as one of these sort of damage repair type therapies, because you're taking these senescent cells that accumulate in your body over time and clearing them out. So I think, while there sometimes seems to be this battle between those two factions, I think often it's not quite as clear cut as, proponents might suggest. And actually, of course, it's also the case that even researching things that you might think only slow down the aging process, and perhaps you don't think that's as beneficial, that can throw up insights into which hallmarks are being affected. It can, what's accumulating more rapidly and more slowly in this case where aging has been slowed down. So it's really not clear to me exactly how we should be apportioning the funding, because we just don't know what's the most exciting thing at the moment.
Yeah, well said. So speaking more about, I guess, like why we age in general, like what would be your main contributors to the aging process? Or do you think it is more a holistic effect of multiple different processes occurring?
I think we're probably going to find that it is a really holistic thing, a sort of massive, interconnected, tangled, chaotic web of nonsense going on inside our bodies, basically. I think it's good to break it down into hallmarks or pillars or, types of damage or whatever it is that you're trying to do in order to come up with some kind of conceptual framework. And I think that we're the results in the lab show that we're going to actually potentially have some quite large successes just going after single hallmarks. I'm really quite excited by the fact that you can give mice senolytic drugs, you can clear out their senescent cells, a single hallmark of aging, and it seems to impact basically the whole of their biology in terms of aging. It seems to turn back the whole of their aging clock. It makes them live longer, it makes them have less disease, it makes them less frail, it makes them less cognitively, can't say that, it makes them less cognitively impaired. Perhaps I'm starting to suffer from that a little bit myself. And it also, it just makes them look great. I was a computational biologist, so I've never dealt with mice in the lab, but mice that have had senescent cell treatment, they just look fantastic. They've got much better fur. So, it's really going after the cosmetic stuff as well, which, unfortunately is going to get certain sections of the population excited, perhaps more so than the medical side of things. But nonetheless, going after this single hallmark has this huge collection of benefits. And it might be that going after, 2 or 3 of the hallmarks or 2 or 3 different processes has a synergistic effect. So, it has an effect that's greater than the sum of its parts. It might be that some things have an effect that's less than the sum of their parts, because you're effectively duplicating efforts somehow. But ultimately, and I talk about this right at the end of the final sort of science chapter of the book, I think the way that we're going to really nail aging is going to be by understanding a sort of systems biology model of it. Because I think that, and you can already see this actually in the discussion around senescent cells. Although senescent cells were first named because they looked like old cells, they were cells that had divided so many times that they weren't able to divide anymore, and that's why they were thus christened. We now understand that senescent cells are a vital part of development. They're used in the developing embryo to fashion certain parts of your body. They're used in wound healing, and that's again sort of a place where you want fresh tissue to grow back, and senescent cells secrete these factors that cause cells to grow. They seem to be implicated in some way in the growth of cancers as well. So again, that's sort of a growth related thing. So it's clear that sort of senescent, clapped out old isn't necessarily the right way to think about them. And you see all these arguments about how do we define a senescent cell. You can say it's a cell that doesn't divide anymore. But is it a cell that's P16 positive? Is it a cell that is positive for the senescence-associated beta-galactose, is it, I think? There are all these various biomarkers that you can...
Yeah.
Galactositis, this is exactly your field, isn't it? So I'll take any corrections you're offering me. So if is it positive for these various markers? I suspect none of these will actually be appropriate for what is a senescent cell, and we'll probably have to come up with some complicated composite marker. And it might be that we end up taking out, the optimal for aging delay might be to take out an entirely different subset of cells, which somewhat overlaps with what we currently call senescent cells, but is actually somewhat something else, and it might be different in different parts of the body. And I just think that is ultimately the way we're probably going to end up solving this problem, because it can't be as simple as, just taking out one particular class of cell and one particular molecule and everything's sorted. Or you feel like biology, evolution might have managed to do that with a little bit more ease in creatures out there in the wild. So I think the ultimate solution is going to be a sort of big holistic systems biology, load of chaos in some ways. But what's more optimistic than that in the short term is that we do have these treatments. You can do interventions in the lab, and they can have dramatic effects on the lifespan of mice, the health of mice, most importantly, even though you only seem to be doing one single thing. So my hope is that, we can sort of use the knowledge that we gain from giving people these sort of laser-guided just senescent cells or just telomeres or just whatever it is treatments. And as we understand how that interacts with their aging more broadly, we can start to intervene in a slightly more intelligent way and hopefully, address multiple hallmarks and eventually all the hallmarks at once.
Yeah, well said. And I completely agree about the whole systems biology approach. And so you've mentioned Senolytics, but what kind of other strategies from obviously writing your book do you think are most promising or seem most exciting at the moment for treating aging?
I think there's a real range, and I'm excited about them all for sort of quite different reasons. The Senolytics, I really feel like are the most exciting in the sort of near term for what I'd call sort of a real hallmarks-based anti-aging treatment, because we've got results in mice that show all these exciting things I just talked about. We've got human trials going on for these drugs now because some of them are pre-existing approved drugs that have been approved for different conditions, so it's something that we can extend into humans. These trials at the moment are obviously for specific diseases where we know senescent cells are a problem, but hopefully if the drugs are effective, so obviously if they help with those diseases, but most importantly if they're safe, we could then think about rolling them out to people who haven't got some such severe problems, and eventually if they're safe enough we could start thinking about rolling them out preventatively. Maybe you're just 60 or 65, so you've accumulated enough senescent cells that it's worth giving you one of these things preventatively. And if all goes well with that, we could easily have Senolytics in hospitals in the next few years for specific conditions if those clinical trials work. And then it's not hard to imagine that maybe 10 years after that, we could start thinking about giving them out preventatively. And that really is, I think, the ultimate sort of paradigm shift in terms of the way that we practice medicine. So that's something I'm very excited about for that sort of variety of reasons I've just mentioned. The sort of near-term stuff that's less exciting in terms of the big picture, but probably more exciting in terms of breaking the ground, are the things like metformin and rapamycin I've already mentioned, these existing drugs that we think might be able to slow down the aging process. And there's this trial that I'm sure a lot of your viewers will have heard of, the TAME trial in the US, where they're going to actually do a proper randomized trial trying metformin to see if it really does slow down the aging process, or if the sort of hints we've got so far have maybe been experimental artifacts. And that, the people who are doing TAME even, they aren't necessarily that, this isn't going to double human lifespan. If metformin doubled human lifespan, we'd know about it already. But the fact is, it's going to lay the regulatory groundwork that allows us, in the past, we've only found it easy to approve drugs that are against specific medical, they call them indications in the jargon of regulation. So specific diseases, basically cancer, diabetes, whatever it is. And if they can lay the groundwork with that study and show regulators that this is a thing that we can do, we can give people, people who would currently classify as healthy, a drug to prevent them becoming unhealthy, and also just show the public and policymakers that we can develop drugs that slow down aging, I think that's a really exciting thing, even if it isn't necessarily going to result in the biggest breakthrough. In the longer term, I find a lot of the sort of wilder stuff that's going on really exciting, and I'm just really fascinated to see how it pans out. And I think the treatment I'd put most in this category is cellular reprogramming, because I often say it feels like it's fallen through a wormhole from the future, because we've got this sort of collection of 4 genes. They were originally discovered by Yamanaka, who was looking for a way to turn adult body cells into a stem cell, and he was interested in those stem cells because they can then turn into any other kind of cell. He wasn't looking for something that could turn back the sort of biological aging clock. But what we've since discovered is that these 4 genes, almost as a side effect of that process, do indeed turn back the aging in those cells, and they do so in a surprisingly, widespread way. They de-age whole, they de-age what's called the epigenetic clock, this measure of how old the cells are looking that way. They can make the mitochondria behave in a more youthful way, and there are just loads and loads of changes that are going on. And now we've even shown that you can give these things to mice, and as long as you only turn them on intermittently and therefore don't cause the cells to go all the way back to becoming a stem cell, you seem to be able to make those mice healthier. So it's an exciting result, but the real question for me, and the reason I say it feels like it's fallen through a wormhole from the future, is can we scratch our heads and find a way to turn this into a therapy that we can actually give to humans in anything like the near term? Because obviously gene therapies are starting to come online now. We've got the first of those being approved in the clinic for specific conditions, for people who are willing to take a punt on something experimental. But I'm certainly not going to be lining up to have the Yamanaka factors injected into me anytime soon. So the question is, can we find a safer way to do this? Can we identify some genes that are perhaps, the Yamanaka factors, they're these skeleton keys inside the cell. They perform such a huge range of changes. Can we find some that are a little bit more targeted and can just address the sort of aging aspect of this without changing the nature of our cells and potentially putting us at risk of cancer and stuff like that? Or, ideally speaking, could we find a drug that can awaken whatever process this is? And, it would be ideal if this could be taken as a pill rather than something more exotic. So the real question with those kinds of therapies is, can we come up with a way of turning them from this idea that works in cells in a dish, works in mice in the lab perhaps, but can we get it to a point where we can consider safely deploying that in human patients? So I think there's this real range of excitement from the very sort of pragmatic near-term stuff to the incredible sort of stuff that's on the horizon. The question is, can we get that stuff on the horizon, a little bit closer to actual practice?
Exactly. And I agree. I also think cellular reprogramming is also really exciting, in particular stem cell therapies. And so you mentioned in the book in a bit more detail about the different potential therapies. And so where do you think we'd see stem cells being used first?
Sorry, say that again?
Why do you think we'd see stem cell therapies being used first, in the clinic, if they ever get safety approval?
I think we're probably going to see them being used again in cases, and a lot of these will be aging related. This is one of the parts of the book where I was more excited because the developments weren't going on so much as a sort of accidental side effect of looking into other things. A lot of the diseases of aging are caused by loss of cells of one kind or another. I think the two things that are most advanced in the clinic at the moment are stem cell therapies against Parkinson's disease. And this honestly, it really shocked me quite how long ago we were doing this. The first stem cell therapies against Parkinson's. So Parkinson's is this disease where you basically lose control of your motor function gradually because of death of a particular kind of neuron in a particular part of the brain. And because it's this very specific type of cell in a very specific localized region, scientists thought, can we replace those cells and thereby, give people a bit of their lives back? And actually, scientists in Sweden first tried transplanting stem cells into Parkinson's patients in the late 1980s. So it's not like this is some wild cutting edge technology that, we just developed last year. This has got a really, really long pedigree. And what they found was, that, they were doing these experiments using cells that were extracted from fetuses, and they had to do this incredibly complicated procedure, pulling out this tiny little ball of cells, I think smaller than the size of a pinhead, from a fetus that was maybe a couple of centimeters long at that stage in development. Then they'd culture those in the lab and inject them into Parkinson's patients. And it was a very, very small number of people, right? It was a handful of patients, but they showed these remarkable results. And there's been this sort of ongoing controversy because that number of patients was so small. It was then some labs in the US attempted to replicate it, and they didn't quite manage. And the labs in Sweden hit back that they hadn't been doing the technique correctly, and therefore that was what the problem was. There's clearly still a bit of excitement about this because there's a massive study going on in Europe at the moment across different hospitals all over the continent, trying to replicate these results again in a much more rigorous way across a whole variety of different locations. And actually, I think the results of that are due out pretty soon. And not only that, at the same time, we've got this huge advance because of that discovery by Yamanaka that we can make these pluripotent stem cells. We don't have to, go on this sort of scavenger hunt through tiny little fetuses that's incredibly complicated. And obviously, some people have ethical problems with. We can generate these cells from other people's adult cells or even the patient's own adult cells and turn them into whatever kind of cell we want, including the cells that are lost in this particular part of the brain in Parkinson's. So I think that, potentially, obviously, stem cell therapy feels like it's something that's been on the horizon for quite a number of years now, but it really does feel like for Parkinson's that's starting to come together. And the other place that I think there are some really advanced trials is looking at age-related macular degeneration. So that's the most common form of blindness in older people. It's when your macula, which is part of the retina, the back of your eye, starts to degenerate as you get older. And again, there are some really cool stem cell experiments going on in Japan. There was this incredibly detailed a very practical safety study that was done in pigs in the US, and pigs are just chosen because they're quite anatomically and biologically close to us in this particular situation, where they tried extracting stem cells from genuine old donors, so old human donors, to make it as realistic as possible. And they went through the whole process as though they were doing a human AMD treatment. The point being to check that this thing is absolutely safe, that there's no risk of something like cancer coming up when you try and modify the genes inside these cells in order to turn them into, back into cells for the back of the eye. And that seemed to pass with flying colours. So again, there's just, it's a really big job, I think, getting something, particularly something biological. You've got to nail down so many different things before you feel confident sticking that into a human being. But I think those are the two that are probably furthest ahead at the moment.
Cool. And so you mentioned Parkinson's disease, which is, I guess, in general, categorized as a neurodegenerative disease. And another really big neurodegenerative disease associated with aging is Alzheimer's. And in your book, you mentioned actually a couple of examples where there's some really exciting research being categorized to treat Alzheimer's. As I was just wondering, could you elaborate on some of those strategies?
That's a really fascinating thing. And I wonder if we're going to at least partly cure Alzheimer's, or not cure, but defer Alzheimer's, I should say, by accident. Because I think that there's been obviously this enormous focus within the Alzheimer's field on one particular kind of accumulated protein, this stuff called amyloid beta. And for the last 20, maybe 30 years, scientists, this is how Alzheimer's was actually first identified, with these plaques and tangles inside people's brains in postmortems. And, so it was obviously thought there are these things that are obvious if you cut a brain open. Perhaps this stuff that's an obvious biological, change that you can see if you're a pathologist, maybe that's what's driving the disease. And so, I mean, the other thing that really, really motivated that hypothesis in the 70s and 80s was the discovery of people who've got what's called early onset Alzheimer's. So that's a form of the disease where you have a particular genetic mutation in your amyloid, in your A beta production, which means that you produce a lot more of this amyloid, and you can get diagnosed with Alzheimer's, I think, even as early as your 30s, but more typically in your 50s and 60s, which is a long time before the age-related form of the disease would normally kick in. So that's been a really, really dominant theory, and we've spent an awful lot of research time trying to clear out this amyloid beta, to try and basically clear out what seems to be waste inside the brain in order to try and sort out dementia. And although we've got very, very good at clearing the amyloid, unfortunately, that doesn't seem to have translated to improvements in anything about patient outcomes. People aren't getting cognitively better or anything like that. So that's, on the one hand, a biological success, but a medical failure. So now there's just a whole plethora of different strategies. I think that the failure of this hypothesis, and the conclusive failure, because so many drugs have failed to improve outcomes, has meant that a whole different range of hypotheses are being explored. And actually, I think a serious one is looking at something like inflammation, so this sort of increase in inflammation as we go through our lives. And it's cellular senescence, again, appears to be at least somewhat implicated in dementia. I just think that quite a lot of these other approaches, although dementia is certainly worth studying in and of itself and trying to uncover what exactly the causes are, I think that by improving all kinds of things in our aging bodies, and we were talking about lysosomes earlier as well, improving the clearance of the lipofuscin from inside lysosomes, which also seems to be a really important factor in all neurodegenerative conditions that we've looked at. I don't really know what's going to happen with Alzheimer's. I think it's really up in the air at the moment, but I do feel like it's such a disease of aging, right? So we already said earlier that the risk of death doubles every 8 years or so in a human being. Whereas dementia is actually even more extreme than that. Your risk of dementia is basically zero unless you've got one of those early onset forms that I mentioned earlier, before the age of 60. And then it doubles every 4.5 years after that, so substantially faster than the rate of aging. It really is an incredibly strongly age-associated disease. So hopefully, by going after some of the other hallmarks, my fingers are crossed that we're going to at least defer dementia by doing that.
Cool. No, that's really interesting. So another area of the field that I find quite interesting at the moment is more the personal approach, and how obviously we're all very different and we're going to respond differently and might need different therapeutics. And so one thing that I know kind of links back to some of the work you did as a computational biologist is about using data and machine learning tools to kind of predict the correct treatment options. And so I believe you did some work looking at electronic health records and being able to predict the risk associated with cardiovascular disease. And so I was just wondering if you could explain more about what exactly it was you were doing and how machine learning approaches could be used to help with personalized therapeutics.
Yeah, the idea of that project was we wanted to take advantage of this enormous rich data set, because in the UK we're very fortunate to have the NHS, and that means that literally everybody basically is on the same medical records database. And so we had data from people's GPs, we had data from hospital visits, we had data on operations that they'd had, medications they'd been given, all this different stuff. And what that had been used for in the past was to try and predict cardiovascular outcomes, like you say. So, say you're a doctor, you're sat in your doctor's surgery and someone comes in, and you're trying to decide whether to prescribe them something preventative like statins. Now, statins are a drug that obviously have side effects, and so you don't want to just be handing them out like candy to everyone over a particular age. You want to be handing them out to people who are actually at risk of having a heart attack. And so what you like is a prognostic risk score, they call it. So, basically a score of how likely are you to have a heart attack in the next 5 years. And so the way those are traditionally calculated is that a bunch of nerds basically get together, a bunch of doctors and epidemiologists and scientists, and they might look at this medical records database and say, okay, we think these are the 20 or 30 most important factors. Some stats nerds then go away and turn those factors into a model. And we say, okay, what this can do is this model can show us with a certain degree of accuracy what your likelihood of having a heart attack is in the next 5 years. And then your GP, when you come in and visit them, rather than sort of looking at your history and sort of having to have a bit of a think and try to base this entirely on their judgment, they can type your various scores, or ideally they could take all the blood test results and it would automatically move into this model. And it would then say, okay, your risk of a heart attack in the next year, or in the next 5 years is 5%. And they say, okay, my threshold for prescribing statins is 4%. So here are your statins, be on your way. And what we thought was, there's just this huge opportunity to put machine learning into this situation, because these scientists, this model I just described in slightly hypothetical terms, had actually been made by some clinicians and epidemiologists, and they were using, I think it was just over 30 of these numbers, but there are literally hundreds, maybe thousands of different codes inside these medical records, and so you're just throwing away so much information by using these expert models. And the other thing we wondered was, not only could you use more information, more isn't always better, because sometimes it can lead to phenomena like overfitting, where you sort of get spuriously accurate results that then don't generalize to the wider population. Not only could you have more of these things, you could also potentially uncover things that the experts might not have thought of. And so that's what we did. We used a variety of different techniques. One of them called elastic net regression, which if anyone's familiar with epigenetic clocks, it's actually the same statistics that's used to derive those. And I also used something called random forest, which is just another statistical technique to try and sift through these hundreds and hundreds of variables and come up with the best possible model. And what I managed was, well, first of all, we did slightly beat the clinicians and epidemiologists, so it was a little bit more accurate with all that data. It wasn't sufficiently more accurate for that to be like a huge success, because we beat them by a fraction of a tiny amount. But what I thought was more exciting was that we did all this without having to understand anything, about cardiology. I'm not a cardiologist, as you know. I'm trained as a physicist, so I knew nothing about hearts before we started that project. And yet, completely naive. Obviously, the machine didn't know anything about hearts either. It could pull out the variables that were most important. And some of those variables did overlap with what the experts had thought, things like heart rate and blood pressure. Obviously, they're important as to, whether or not you're going to have a heart attack. But we also pulled out some stuff that you really wouldn't necessarily have expected. And I think my favorite example of that was something that's prognostic of whether or not you're going to have a heart attack is if you've had a telephone consultation with your GP. Now, I guess that variable might not work so well in the COVID era, because obviously all of us, if we want to interact with our GP, have to do it by telephone, or are certainly encouraged very strongly to do so. But back in those days, having a telephone encounter or even a home visit is sort of the more extreme version of that. If your GP has to come to your house, what that means is you're probably so ill that you can't make it to the surgery. And therefore, what that suggests is that, perhaps you're at higher cardiovascular risk. And so this is sort of almost an administrative code that's put in there. It's nothing to do with the patient's actual, health or blood markers or anything like that. And yet we found it was quite predictive. So what this allows you to do then is to uncover these things that can be useful, that can help a prognostic score, but that wouldn't necessarily have been thought of by experts. And I think what's most exciting about that is, as I say, my score didn't do vastly better than that the cardiologists already had. But the fact is that heart disease is a very well characterized disease. We know a lot about the risk factors. If you get a doctor in the room, they'll immediately reel off the 10 things they think are most important. It's fairly well understood. But the fact is, building those expert models, firstly, it requires that understanding. We've just talked about dementia. We don't necessarily have the understanding of the etiology of some of these other diseases. And secondly, there are just loads of diseases. So if you want to do this with some slightly more obscure condition, you might have to get a bunch of experts together, and it takes the same amount of time to build this hand-built expert model. And yet it might only apply to a far smaller fraction of the population. So it might just not be worth doing economically or whatever. So what's cool about the machine learning is whatever the condition is, and whatever the state of the expert understanding of it, you can try and see if the model can pull something out. And if it can, then you've potentially got a prognostic tool for something that you otherwise wouldn't have been able to have. So that was what we were hoping to do. And I think that's why that's so exciting. The question of whether it's going to lead to personalized treatment, I think, is a much thornier one, because the thing with prediction is you've got quite good data, and particularly something like a heart attack or death. Like, it's very easy to find when someone has one of those things. It's quite difficult, you'd think, to dodge, that appearing in your GP record. Actually, intriguingly, one of the other difficulties with using this, it's called sort of found data, when you use a record that already exists rather than a record that was specifically created. I've forgotten the numbers now, but it's remarkable. So we had access to 4 different databases. There was the GP records, the hospital records, a special heart disease database, and there was a 4th one that I've forgotten right now. But it was quite shocking. A heart attack you'd think is very major. You can't fail to go to hospital if you have a heart attack, and your GP cannot fail to receive a letter from the hospital telling them that you've had a heart attack if that happens. And yet there was still some huge percentage of patients who weren't recorded in all 4 of these data sources. And it's just, as I say, this is one of the hazards of using found data. It's often got lots of missing values, and so it doesn't necessarily have everything you want. Nonetheless, heart attacks, death, they're quite unambiguous outcomes, shall we say. Whereas if you're looking for something a bit more subtle, it can be a bit more challenging. And the particular challenge with treatment is that outside of the context of clinical trials, doctors normally prescribe the best treatment for that particular patient in their estimation. It's normally the treatment that's effectively the first line treatment, the first on the list. And they then move to the second one if that doesn't work for some reason, which often might not be not particularly clear. So the question is there going to be a diverse enough range of data in terms of what treatments people were given in order for the machine to learn? And my sort of dream for the future is firstly that just machine learning techniques generally will advance. And I think we're already seeing that. Particularly neural networks are really hot and exciting at the moment, but I guess these techniques are evolving all the time and getting better and better at digesting and making sense of huger and huger quantities of data. But I think what would be really exciting, and this is again particularly applicable to a country like the UK, where we do have a national health service and literally everybody is enrolled into effectively the same system. I'd like to see a lot more adaptive clinical trials happening in a healthcare setting. So quite often, patients have complicated diseases. It's not necessarily 100% clear what the absolute best drug for them would be. And in that case, it would be nice if the computer could randomize them to one of a few relatively good options. And then, over sort of months and years that they watch those patients afterwards, you can then draw that data together. And as it becomes clear that certain treatments are better for certain types of patients, maybe you could increase the probability that a patient with those characteristics gets that treatment. So people are always getting, or the largest percentage of people are always getting the best possible treatment, but there's always a little bit of experimentation going on in the system as well, just to make sure that what we think is the best possible treatment is the best possible treatment. The issue, of course, then is that that's really exciting. And I think, firstly, it's exciting to nerds like me. And secondly, I think it probably would save the most lives overall if it were carefully implemented. But the problem is that clinicians and members of the public, I think, have this real uncertainty about randomization in a treatment context. I think if you randomize in a trial, then everyone's like, oh, I understand why we're doing this. We've got to find out if one drug's better than the other. But by the time you get to the clinic, you want everyone to be getting the same treatment. You don't want to feel like you're missing out on the best treatment. And often that's, in a sense, slightly misguided, because we don't always know what the best treatment is for a particular situation. But at the same time, you don't want to feel like you're the person who's been randomized to placebo, or you're the person who's been randomized to the second best treatment, because that's what the computer told you to do. That makes it feel even worse if there's not necessarily a human making every aspect of the decision. So I think there's a lot of exciting potential for that. But it's just a question of, how can we implement those things in a healthcare setting?
Yeah, no, that was really fascinating. That was amazing detail. So in terms of also the potential of using these machine learning techniques, what kind of measurements do you think are going to be most necessary to have? For example, like blood measurements, or even things like doctor visits seems to also be important. And then I guess on from that, what are kind of like the major challenges to also trying to use this approach more globally? I guess things such as security of the data, or actually trying to pull data from different sources.
I think in answer to the first thing, we just don't know what are the most important things. And we can probably make some reasonable guesses in terms of, doctors can obviously tell you with more wisdom than I can what are, considered to be important blood tests. But at the same time, you don't necessarily want to always go with what doctors think are the most important things, because, though I massively respect doctors for the ridiculous amount of knowledge they hold in their heads about human bodies, there have also been a lot of times in the past when what seems to be the medically obvious particular answer has turned out not to be correct at all. So I think we have got to sort of try and throw a bit of randomness in there and sort of let the machine learning models do their thing. In terms of the data security, yeah, that is a huge challenge. But I think that's definitely something we're all waking up to, not just in medicine, but in our everyday lives as well, because, we've got Facebook and Google and all these different companies that are just basically, their business model is to aggregate as much data about us as possible. And that's really starting to include health data as well, because Google is starting to do work. They've been doing machine learning on medical records data as well, because it's clearly, a huge business opportunity as well as one for improving human health. Luckily, I think because there is that awareness, we are starting to get a bit better at this. And actually, COVID here in the UK has driven some really big innovations in this area. There's a sort of family of trials called Open Safely. And I think that's some tortured acronym, but I'm afraid I don't know what for. But the idea there was that this was actually, as I say, specifically to look at COVID. They were trying to find out which patients are at highest risk based on their medical records, based on hospital admissions and who comes into hospital with COVID and so on. And the way that that system was set up was that everybody's medical records were stored on an entirely secure central database. And then if you wanted to analyze those records, then what you had to do was submit a job, which was then processed on this central database, and then it would spit out the answers. But you never actually saw, you never had your hands on the actual data. And so that's just a really great model, because then what that means is that, researchers can effectively do this kind of stuff almost entirely anonymized. The real challenge is that actually, although we deal with anonymized data, it's sometimes called pseudonymized, because, you don't actually, it's very hard to truly anonymize stuff. So when you say pseudonymized, it might mean that it doesn't have your name, it doesn't have your actual date of birth, we might round it to the nearest month. It doesn't have your NHS number, for example. It's got some random identifier that we then give, we then give the patients. The problem is that even if you hand over what seems like a really well pseudonymized data set, it's actually remarkably easy to start de-anonymizing people to work out who these individuals actually are. Because if you think about the number of unique characteristics we each have, obviously you've got your gender. Even knowing your month of birth is a good start. Knowing which part of the country you live in, you can start to isolate people very quickly, particularly if they have a rare condition or something like that. You might be able to identify them down to just a handful of people in the UK just because of one particular disease that they've been diagnosed with. So there are definitely challenges there, but I think we're hopefully getting better and better at navigating them. So it's watch this space, I guess.
Yeah, that's really interesting. And so you mentioned elastic net regression models that you use for your studies, and how that's also used in epigenetic clocks. And so I was just wondering if you could first maybe comment on your opinion on epigenetic clocks at the moment, and then maybe just discuss a bit further, like where else do you see the use of maths in trying to treat aging?
Yeah, I actually think, to answer the second part of your question first, I think that biomarkers are one of the most exciting places that maths and machine learning can come to bear, because we do have all kinds of data sets. And I think a really cool example of this actually is just looking at people, right? So there was this fascinating study that came out in 2009 where people were asked to rate how old they thought other people looked. And what they found was that people who were rated as looking older seemed to actually be biologically older than their people, other people the same age who looked younger. They got more diseases, they died more quickly, and so on. And since then, we sort of tried to formalize that a little bit by using machine learning. Take a photo of someone's face, and then the computer will analyze the various things on the image. And obviously, it being machine learning, we can never be quite 100% sure what it is it's analyzing. But it'll then spit out a number that tells you how biologically old you are based on that. And that's a really cool application, because rather than taking loads of blood tests or doing a full body MRI scan or however else we could get huge volumes of data to put into these models, you can just take a photo. And actually, there was an example of trying to do this for mice as well. And again, if you're doing a mouse experiment, you can take all, you can measure their epigenetic age and do all this, complicated blood tests, or you can just take a before and after picture. And if you could get that to work with sufficient accuracy, that's a huge improvement. So I think that's a really cool place that we can use maths. In terms of the epigenetic clocks, I think it's sort of a case of waiting and seeing, unfortunately. They're clearly very good at predicting the biological age of people who are living in our current normal environment. But the problem with all machine learning, or a lot of machine learning, is it's often looking for correlations and not what's actually causative. So it might be that if I've got this particular spectrum of values of blood tests, and that can then be used to derive an epigenetic clock, and that then tells me that I'm this old. You see people trying to optimize a particular blood test value, but that's kind of cheating, because is it that blood test is actually the thing that's driving your aging, or is it that whatever value it's measuring actually is indicative of a whole range of other things going on in your body, or that value only makes sense in combination with the other values that have been put into the biological aging clock that you're looking at? And what I think we really need to see to get properly excited about this stuff is if we do some therapies that we know slow down the aging process, and then the epigenetic clock responds in a commensurate way, then we can say that we're measuring something meaningful and not just measuring, some aspect of the way that we currently live our lives effectively. And there are some early signs that this works. So you see that in animals that are undergoing calorie restriction, for example, their epigenetic clocks seem to tick more slowly. And there was even this trial of thymus rejuvenation, which showed that people's epigenetic clocks were reduced by, I think it was a few months or maybe a year after the treatment had been done. So I think it's really exciting, but it's just a question of watching this space and trying to find out, trying to find out if these things actually do show us what we need them to show us. Because at the moment, the ideal way to do a trial, and the way the tame metformin trial that I talked about earlier is going to be done, is they're going to give a bunch of people, I think it's between the ages of 60 and 79, metformin or a placebo, and they're going to watch them for a few years. That trial has to be big, because ultimately, even a 65-year-old only has a 1% chance of dying that year. And that means that, in order to actually see one 65-year-old die, you need to have 100 65-year-olds in your trial. And that means if you want to see a difference between the treatment and a placebo arm of the trial, you have to have hundreds and hundreds of people involved. And you have to watch them for several years in order to get good enough statistics to actually draw conclusions. Whereas if we could do epigenetic clock measurements, for example, you could take a blood test from everyone before the trial started, you could give them metformin for 6 months, and then every person in your trial is a data point rather than just the handful who died. Because everyone has an epigenetic age before, and they've got an epigenetic age after, if I can say that. And that means that everyone's a data point. So in a far shorter time, with far fewer participants, potentially, you can come up with the same results. And that's obviously really important for us as aging researchers, because we don't want to be waiting 10, 20, 30 years for the ultimate clinical trial to find out if giving Senolytics to 50-year-olds is something that's going to slow down their aging. It's far more useful if we can get results in months or small numbers of years. And the smaller the number of participants you can use, the less expensive these trials are, so the more of them you can do in parallel, the more stuff we can try out. So I think it's really exciting. It's just a question of, are they going to work as we hope they're going to work?
Yeah, exactly. And so evidently you're very good at communicating science. And I believe now that you do science communication as kind of like your full-time career. So I was just wondering if you could maybe talk about some projects that you're working on, or what is it you kind of get up to on a daily basis besides talking to people like me?
Yeah, it's a good question. It's a very strange job, this. So I spent a lot of time writing this book, right? So that's I was fortunate enough to get a book deal toward the end of my second postdoc, and basically decided to write full time. And I'm so glad I did, because it effectively took me 2 years full time to write the book. And it's just because aging biology is such a huge field. It obviously touches on every aspect of biology, and so it's not possible to be an expert in all of those things before you start, and I certainly wasn't. That meant I had to talk to loads and loads of scientists. I had to read loads and loads of papers, and I wanted to make it really, really robust as a book as well, because I've tried, as you sort of mentioned earlier, to make it as accessible as possible to as many people as possible. But at the same time, I wanted, if you're a biology nerd and you're thinking, oh, this all sounds a bit sci-fi and kooky, there are hundreds of references in the back of the book where you can trace up everything I've said and make sure that I'm not talking total nonsense. And it was really, really important to me that it was as scientifically robust as possible, so it couldn't just be dismissed out of hand as some sort of fringy theories being passed around. So that was the first thing that I've spent an awful lot of time on, is just writing the book. At the same time, I've been trying to make some YouTube videos to talk about various aspects of science, not just aging biology, because I think it's really important to try and engage people on a variety of different cool scientific things. And I mean, I guess ultimately one of the reasons why I want to live a longer, healthier life is to have more time to explore this wonderful universe we find ourselves in. I'm still, that astronomy bug that I had back at the age of 7 hasn't entirely gone away. I've also been writing articles for newspapers and doing a lot of press often around the book. And that's just been a really, really cool opportunity. There's some talk of maybe a TV series, but these things are totally, totally unknown. I've done a bit of TV presenting on other things. So it's just a very strange sort of collection of stuff. I guess I'm starting to think about a second book now. So I guess we'll just have to see.
Awesome. No, that sounds really exciting. And I think I saw you on the Russell Howard show recently, which is also super exciting.
Yeah, it's really cool. That's definitely a sort of childhood dream of mine. I've always been fascinated by comedy. So to be on a sort of comedy news, topical satire show is absolutely fantastic. And also what's really cool about that is I'm genuinely passionate about getting the message out about aging to as many people as possible. And so it's all about, going to these really mainstream outlets, be it mainstream news shows or comedy shows or whatever it is, and just telling them about this stuff. So it doesn't seem fringe and weird. It's obviously, I love talking to scientists. You can probably tell, I'm happy to nerd out about anything at any level of detail. But I think the thing that really excites me is talking to people who haven't necessarily been exposed to these ideas at all before and showing them this is mainstream exciting stuff.
Exactly. And so talking more about generally just aging and health in general, I know obviously a lot of these treatment options, there isn't enough data to talk about them or implement them. But in general, what kind of advice would you give to someone who is trying to be more healthy?
There's a health advice chapter in the back of the book, and I think it falls kind of into two categories. One of them is the stuff you've probably heard 100 times before, but I hope that understanding a bit more about aging biology will really encourage you to stick to that advice. And that's the sort of a big part of the sort of my health regimen has been driven by that. In that, I knew that you're supposed to, not eat too much and try and eat a balanced diet and try and get some exercise. And obviously not smoke is the big thing that should be in, red flashing lights, that you really, really shouldn't do these things. The thing that really encourages me to, watch my diet a bit and try and make sure I do get plenty of exercise is that these things literally slow down the aging process. So if you think about, something like exercise, it's obvious when you exercise that you can feel your heart beating in your chest, you can feel your lungs going, you can feel your muscles being worked out. It's clearly a cardiovascular workout. It's clearly helping your muscles. But as you understand more about the biology, you realize that it's going to be, decreasing inflammation throughout your body. It doesn't just decrease the risk of heart disease, which is the sort of obvious one. It decreases the risk of quite a few different kinds of cancer. It even decreases the risk of dementia. So it really suggests that, again, just like Senolytics in a way, you're intervening in a whole range of different aspects of the aging process. And that makes that seem really, really cool to me. The other thing that I find is that there are some bits of health advice that you just wouldn't expect but for aging biology. And I think my favorite example of that is trying to brush your teeth. So it's been noticed throughout the last sort of 10, 20, 30 years that people with worse oral hygiene tend to go on to have worse heart outcomes, worse cardiovascular problems. And you might be sort of scratching your head and thinking, well, this is another example where correlation doesn't equal causation, because maybe it's that people who have more time to take care of their teeth, they have less tooth decay, they have less gum disease, but also they have more time to exercise, they have more time to cook really good meals, they have more time to take care of their health generally. And so these two things are related, but they're not causally related. But actually, as you start to unpick the biology a bit more, you realize that this is again a source of that chronic inflammation that I've sort of alluded to a few times. Because in youth you have this inflammatory process. It's very, very important. It's the way that your body responds to external threats. So if there are bacteria in your mouth, you'll get inflammation. Your immune system will rush to your mouth and try and clear out those bacteria. The problem is, of course, that your immune system can never quite get rid of all those bacteria. That's why dentistry is in a way so archaic, because they literally drill bits of your tooth out because there's no other way to get rid of the bacteria. So what happens is there's this sort of constant fizzing going on inside your mouth of inflammation. And so the inflammation goes from acute, which is like turns on very rapidly when there's a problem and turns off very rapidly when that problem's finished, to chronic. So it's sort of constantly going on. There's sort of a paranoia in your immune system. And we know that that can drive the whole aging process. And in fact, that seems to be the causal link between having clean teeth and having a well-functioning heart. There's even some evidence that, again, it could slow down dementia. So, if there wasn't enough sort of incentive to avoid dental bills and to avoid painful tooth decay, to keep your teeth cleaned, to floss and brush your teeth every night as thoroughly as possible, then I think the fact that it can literally slow down aging is something that's really encouraged me to keep on top of my dentistry.
That's really good advice. I'd say more generally, do you have any advice for those wanting to kind of get into the field of aging biology? Obviously, you've had a kind of more unconventional approach, but do you have any, just general advice about having a scientific career?
I think it's just important to sort of have your goal in mind. It would be ideal to have your goal in mind from the start, right? Perhaps don't do the physics PhD, but at the same time, don't feel constrained by your qualifications so far, or whatever you already feel like you've got expertise in, because it is possible to make these career switches at any stage of your career. Don't feel constrained by your A levels or your degree or your PhD even, because you can make these changes whenever you want. And I think it's also important just not to be disheartened, because one of the real challenges of aging biology, I sort of mentioned this earlier, there just aren't that many labs doing it. So it might be that you can't find your perfect master's project or your perfect PhD working, in exactly the group that you wanted, studying exactly the aspect of the aging process you wanted. But the fact is, a lot of the knowledge that you acquire, maybe, as I say, I wouldn't recommend a physics PhD, but if you go and do some research in an excellent cancer research lab, and you learn loads of lab techniques, and you learn, pipetting and molecular biology, of which I cannot possibly go because I've never worked in a biology lab, but all those techniques are incredibly cross-applicable. If you become an expert in a certain particular kind of microscopy, then that microscopy is probably going to be applicable to the aging process as well. So by the time you get to the level where you're applying for your own funding, you can start to move your research, sort of segue into that direction, which means you could perhaps carry on doing whatever your core expertise is, and make sure you can keep on churning out those papers, which is so important for the academic career, but at the same time start to redirect a bit of your energy towards aging biology. So basically, I think just keep your head down and carry on as best as possible. If you can get a position directly working on exactly what you want from the beginning, that's great. But if you can't, just press on, keep gathering expertise, and eventually you'll probably be able to find a way to apply that expertise to aging.
Exactly. So where can people go to find out more information about you and the work that you do?
So you can check out the book at ageless.link. I've got a website, andrewsteele.co.uk. I'm also a lot on Twitter, which is @Stato. My YouTube channel is Dr. Andrew Steele. I like to keep all these things different just to keep everyone on their toes. And I'm on a variety of other social media stuff as well. So if you just search for me, you'll find me there.
Okay, great. I'll put some of those links into the description as well. So Andrew, it's been great to have you on today. I think we've covered so many different topics, and it's been really informative. So just thank you very much for the work that you're doing.
Thank you so much for having me. It's been a lot of fun.
Thank you. So I hope you've learned something in this video. Thank you to my Patreon supporters, and thank you for listening.